Hello, in this video we will discuss about the whole process of the fertilization. So first of all we should understand about the overview of the fertilization and transport of gamete and fertilization we will discuss. So that was the past video which that is I am attaching together to understand better. So here is the basically I am drawing the female reproductive system which that's contain a vagina for penetration of the sperm during the mating and the cervix as well as a uterus. Uterus and cervix and vaginas contain a smooth muscle during the orgasm. The muscle contraction will lead to uptake of the sperm. So here is the fallopian tube. This fallopian tube is contain a cilia and smooth muscle which that is propel the oocytes and cumulus complex toward the uterus. So it means during the fertilization the fallopian tube is play important role for the fertilization because the fertilization is occur in the fallopian tube. So here is the sperm. We will discuss first the 150 to 600 million sperm into the vagina of a female at the time of one ejaculation, and sperm reach the ampullary portion of the fallopian tube. So the uterus basically is the fallopian tube is sweep over the ovarian surface and pick up the oocyte and surrounding by its complement of the granulosa cell. So important thing is that the fimbriae are present in the beginning of the fallopian tube, which that is also used for the sweep of the egg during the ovulation. So here is the fallopian tube is basically sweep over the ovarian surface and fimbriae also and pick up the oocyte surrounding by its complement of granulosa cell. So the sperm is able to fertilize, so that is known as the capacitation in epididymis. So this thing is the the maturation of the sperm are present in the testes. Basically the sub part of the testes contain a epididymis. So that is here is the testes I am drawing to understand where the meiosis will be occur. So here is the seminiferous tubule which that is interconnecting to each other is looking like this, and this is the seminiferous tubule play important role for the meiosis. So the spermatids formation. So this spermatids is the secondary spermatids basically is formed after the meiosis, and that's stored into the basically is the initial segment. So initial segment to move into the caput and basically first is enter into the rete testis. This rete testis is here and go into the corpus, and epididymis will store and mature, and chorda is used for the releasing of the sperm. This is the cross-sectional view of the testis. So here is the capacitation of sperm is the stored in epididymis, and capacitation will be occur and modification of protective protein coat from the sperm cell membrane appears to be an important molecular event in the process of capacitation. So that means the capacitation is very important before the fertilization. So after the capacitation, the fertilization will be occur due to the ability of the sperm to for fertilization. So here I am drawing the sperm structure, which that is the whole mature sperm, which contain a head and midpiece as well as flagellum. First of all, I want to say that the 36 degrees Celsius to 35 degrees Celsius temperature is required for the maturation of the sperm. So the maturation of sperm near to 36 to 35, this temperature controlled by a hypothalamus. So our body temperature is 37 and maximum is 38, but during the fever it can be increased, but the normal is 37. So it means Below our body temperature, the testis is required temperature for the maturation of the sperm. So here is the sperm structure, which contain a head and midpiece and flagellum. Flagellum is used for the movement, and mitochondria are present in the midpiece, which that is provide the energy for the movement of the flagellum. And the centriole is present, which that is not involved in the in fertilization. And here the nucleus contains 23 chromosomes in the form of haploid. And here is the acrosome, is contain a digestive enzyme. This acrosome, when insert during the fertilization, is used and play important role. So here is the egg, which contain a zona pellucida here inside, while upper side is the known as the corona radiata, which that is made up of cells, basically uh, subcells. So here is basically when sperm first enter, acrosome bind to zona pellucida before moving inside corona radiata and produce the acrosomal reaction. And the second one is the acrosome reaction will lead to enter and penetrate this head into the zona pellucida. After this, when enter into the zona pellucida, that protein will be digest and lead to enter the basically nucleus inside. So third one is that. And after the entering of the nucleus of the sperm, and the other part of the sperm will be left behind. And this is the nucleus of the egg and female egg, and the sperm of the male nucleus, which that is combined together, is known as fertilization. So this fertilization lead to become a zygote. This zygote is the diploid. So here the sperm nucleus enter the cytoplasm of the egg, and here is a calcium increase inside, and oocyte triggers the cortical reaction and harden the membrane. After the harden of the membrane, so the fertilization membrane will be inhibit the further sperm entry. So that is prevention of the polyspermic action. So all these things was the overview. So let's begin to understand with the whole detail for the fertilization of the sperm with egg. So you can see that here is basically is the I am drawing the uterus, which that is contain fallopian tube, ovary, fimbriae for sweep the egg during the ovulation, and the cilia allow to propel the egg in the fallopian tube. So here you can see the reproductive system of the female. So here the fallopian tube, and here the cilia allow to propel the egg. And here inside is, you can watch that uterus allow to implantation here of the egg, zygote. So basically when fallopian tube will allow to move the egg during the ejaculation of the sperm during the mating, the vagina cervix allow to penetrate penis, and first sperm enter from vaginal tract. So during the orgasm of the female, the smooth muscle contraction to allow the uptake of the sperm. And here is the ovulation is the release of egg from the ovary. You can see here. So that's all thing is due to the sympathetic and parasympathetic nervous system, which that is before the orgasm, that is a parasympathetic nervous system, while that during the orgasm is the sympathetic nervous system will be activated. Anyhow, now we should understand about the whole mechanism and the process of the fertilization with the detail. I hope you make sense about the overview of the fertilization, but here we can understand that the whole process of the fertilization one by one. So I will cover the 8 step to understand the fertilization. So here's the number 1 that we can here is the basically a brown color shows that the vitelline envelope, while the upper side is known as the zona pellucida contain glycoprotein. So this zona pellucida is contain glycoprotein, and this protein is the ZP protein, Z protein. and Z protein 1 cross-link the filamentous ZP2 and ZP3 into a lattice. So three type of Z protein, which that is used, and it is due to the zona pellucida protein. Zona pellucida 1, zona pellucida 2, and zona pellucida 3. So here is the follicular cells. This follicular cells is basically is the corona radiata. And after this entry of the initial number 1 is the signal transduction cascade will be occur, and the follicular cell will be sidewise and the sperm will be enter and push inside, and that's contain a receptor. This receptor is known as the ZP3 receptor, which that is bind with the protein ZP3 are present in the zona pellucida. And after this binding of the ZP3 protein, basically ZP protein are present in the also a white line envelope. So here is the number 1, and the second one is the number 2 is the acrosomal reaction. In this acrosomal reaction, during the acrosome reaction, an increased acrosome contains hydrolyzing enzyme for uh, for penetration in zona pellucida. Acrosome is unique organelle basically, which that excrete enzyme. So this means here is you can see that is basically the acrosomal reaction. Before the acrosomal reaction, the calcium will be released from the sperm into the zona pellucida. During the calcium releasing, so the ZP3 receptor is dissolved, and the basically is the rise in calcium inside the sperm cell basically trigger the exocytosis of the acrosomal reaction. So the calcium is basically inside is the release from the organelle, for example from the vesicle. So the acrosomal reaction which contain hydrolytic enzyme, which that acrosomal content released by exocytosis. So this basically is the calcium will be rise inside of the sperm, which that is lead to produce the acrosomal enzyme is known as the hydrolytic enzyme, which that is hydrolyze the zona pellucida, and acrosomal content released by exocytosis also from the from the acrosome. So basically exocytosis and calcium is also release also. So here is a white line envelope are present is here, which contain a receptor. So receptor is also present in the plasma membrane. We will discuss also. This receptor is also present in the plasma membrane. So the zona pellucida is dissolved, and the sperm bind with the isomu. Basically, the sperm bind with the isomu. Isomu are present in the sperm, while the sperm binding receptor are present in the vitelline envelope is known as the juno. So the number 3 step is the hydrolytic enzyme contained in the acrosomal cap are released. The enzyme dissolved into the zona pellucida. So after the dissolving of the enzyme, it means the zona pellucida is also dissolved and hydrolyzed. And after the hydrolyzing, the bind with the isomu with the juno, and basically the isomu with the juno binding is basically look like this. And the sperm binding receptor is juno, and the protein are present in the sperm head from the acrosome is isomer. So after this binding, this plasma membrane basically inside is the plasma membrane is also contain that receptor, and this after the binding with the receptor, which that is breakdown of the plasma membrane, and the sperm nuclei will be released. How it's possible? We will discuss in this manner. So here is the basically the number 3 is the basically the plasma membrane is basically inside, and the cortical granules are present, and sperm binding receptor basically, and stored calcium inside. and the cortical vesicle which that store. During the releasing of the cortical granules which that store in the cortical vesicle and store calcium uh, in this time. So when released, this basically is the deposit into the between the vitelline and plasma membrane. So this deposition of the calcium, so we will discuss about this, and basically is the releasing of the cortical granules will lead to calcium rise inside oocytes and trigger the cortical reaction and harden the zona pellucida by production of protein from vesicle. So you can see in this diagram, number 4 is with the head of sperm no lying sidewise and micro and basically the microvilli on the oocyte surround the sperm head. The two membrane fuse. So here is the two membrane will be fused basically with the sperm and the egg membrane and release the basically nuclei. So the calcium rise inside oocyte, trigger the cortical reaction and harden the zona pellucida by production of protein from vesicle. So the PLC zeta protein is basically is released from the nuclei, and this release to stimulate the calcium from the organelle which that is store the calcium in the exocytosis, and the calcium will trigger the zinc spark. This zinc spark will be released and deposit into the between the plasma membrane and vitelline membrane, which that is basically deposit and stimulate the number 6 is the calcium rise inside and oocytes. The second meiotic division will be induced and formation of second polar body, and depolarization of plasma membrane will be occur. So these two polar nuclei will be formed after the second meiotic division. So the second meiotic division will be produce a second polar body, and the two polar body will be deposited in this site. And after the deposition, so this mechanism is here is you can see that the incomplete second meiosis will be complete and produce the one more polar body which that is deposit in it between the vitelline and plasma membrane, and basically the pronuclei are male and pronuclei are female, which that is after the second meiotic division by the stimulation of the calcium rise inside the oocyte will be bind, and basically will be bind the two haploid gamete into the diploid zygote, which that is diploid zygote formation we will discuss in this 7th step, and harden the zona pellucida by changes of chemical composition. Why? Because the calcium deposition as well as the another protein, the PLC zeta, which that stimulate, and the other chemical changes which that is lead to harden the zona pellucida. Why? We will discuss also. So here is the 7, and harden the zona pellucida by changes of chemical composition, and male and female pronuclei will be fused. And the 8th step is the fertilization. And the membrane zinc spark also help to deposit proteins such as collagen, calcium deposition for prevent to polyspermia. So it means the zinc spark is important for the deposition of the collagen in the zona pellucida for the hardening of the zona pellucida. Why? Because it is inhibit the more sperm entry. So it means the fertilization membrane will inhibit further polyspermia, and polyspermic process is the disorder basically which that is unable to normal grow. So that was the process of the fertilization, which that is the 8th step. So after fertilization, what will be happen? After fertilization, basically first of all here is I am again discussing about the fertilization when sperm will be enter. So one sperm will be enter and fertilization to become a zygote. Zygote is basically the diploid, while because the number of chromosome half is come from the female but the half is come from the male, and the oocyte is basically the female and sperm is the male gamete, which that's contain half number of the chromosome. And it means after the formation of the zygote, that is the number 2 is the cleavage formation. Cleavage formation is the basically mitosis, which that is the cell division, and 2 cell stage to 8 cell stage basically, and 2 cell to 4 cell stages after the cleavage formation, and 8 cell stages to become a morulation. And that's morulation is the morula formation. Morula is the basically is the mulberry like structure. Basically the mulberry plant which contain a fruit is known as a mulberry, basically is look like that, and that is why it is known as the morula, because it is belong to the family Morasalba, which that is belong to the family Moraceae. So that is a multiple same type of cell packages is look like this. After this, basically after the morula formation in the fallopian tube, so form the blastula. So this blastula, if we will talk about the cross section of the blastula, that is blastocele are present inside, that is developed, and that blastocele is contain a basically fluid. After the blastocele in the recent blastula formation to become a blastocyst when the expand of the blastocele. After the blastocyst, basically blastocyst is able to implantation of the uterus, it is known as blastocyst. So remember about that, blastocyst is important for the implantation. So that when implantation into the endometrial wall endometrial wall of the uterus, so that is known as the blastocyst. Otherwise it is not known as the blastocyst. So here the fallopian tube will lead to go into the uterus, the endometrial wall, and implantation of the blastocyst. So that is here is the uterus. So I hope you make sense about that video. That was a long video, but I will explain too much. So I hope you make sense about the fertilization and the process. So the next video we will discuss about the superfecundation and the twins, identical twins.